Welcome students. I am Dr. Maria Signola Fernandez, Assistant Professor in Botany, St. Joseph Vaz College, Kortalim, Goa. Welcome to the program Bachelor of Science first year. Subject Botany, Semester 1, Course Code BOC 101. Course title Biodiversity 1. Under that we have microbes, algae, fungi and bryophytes. Title of the unit is Bryophyta. Module name Anatomy of Funaria. In today's module, we will study about the internal structure of stem, internal structure of leaf, internal structure of capsule of Funaria. By the end of this module, you will understand the simple internal structure of stem, leaf and capsule of Funaria. Students, we will study now the internal structure of stem. The stem reveals a simple internal structure. If you see in the diagram, you can see the cells, they are arranged in three distinct zones. The first zone is the epidermis, followed by the cortex, and at the center we have the central cylinder or the central strand. The epidermis, it is the outermost single layer of cells external to the cortex. The cells are small and they contain chloroplast in the younger portions of the stem. In mature stem, the epidermis is thick-walled and lack chloroplast. Stomata is absent. The next layer is the cortex. Cortex is surrounded by the central cylinder and is relatively of greater thickness. It is composed of undifferentiated, large, thin-walled parenchymatous cells. In younger stem, the cortical cells contain chloroplast. At the maturity, the cortex is differentiated into an outer, thick-walled cortex and an inner, thin-walled cortex. The outer cortex is thick-walled and at maturity it turns in brown color, so it is also known as brown cortex. The inner cortex lies next to the central cylinder. The third zone is the central cylinder or the central strand. It is termed as central cylinder or central strand because it forms the core of the stem. The cells consist of vertically elongated, thin-walled, narrow, compactly arranged without protoplasm. These thin-walled dead cells with non-lignified walls are hydroids. The central cylinder provides mechanical support and even functions in the upward movement of water and solutes. Students, now we will study the internal structure of leaf. Leaf consists of well-defined midrib with two lateral wings. If you see in the diagram, you can see there is a well-defined midrib and with two lateral wings. The cells are composed of single layer of undifferentiated parenchymatous cells. They are comparatively large and prominent chloroplast. They have comparatively large and prominent chloroplast, so they can function for photosynthesis. The midrib has an upper and lower epidermis. There is an upper epidermis and there is a lower epidermis. Below the upper epidermis, a small group of thin-walled large cells are present and thick-walled small cells with narrow lumen is also present. Students, now we will study the internal structure of capsule. The capsule is highly organized structure of the sporophyte. If you see the diagram, the cross section of Funaria capsule, it is a pear-shaped and asymmetrical. Internally, it is divided into three distinct parts. First part, that is the sterile basal part, is the apophysis. The central fertile region is the theca region, and on top you have the operculum or also known as lead. We will now see what is apophysis. Apophysis, it consists of thick-walled epidermal layer. Stomata are present in the epidermis. Below the epidermis is a broad spongy zone of sterile cells rich in chloroplast. The innermost tissue is the central conducting strand and is composed of vertically elongated, narrow, thin-walled cells devoid of chloroplast. The second region is the theca region. This region of capsule is highly differentiated and it is having several cells in the thickness. The outermost layer is the epidermis with few stomata. Below the epidermis is two-layered, compactly arranged parenchymatous hypodermis. Within the hypodermis is the 1-2-layered, loosely arranged, spongy cells with chloroplast. Inside the capsule wall is a wide, cylindrical air space crossed by strands of narrow, elongated green cells called the trabeculae. Now, the trabeculae, they connect the innermost layer of the capsule with the outer wall of the spore sac. The center of the theca region is a solid cylinder of compact parenchyma cells, and these are known as columella. Surrounding the columella is a barrel-shaped structure which consists of inner spore sac, which is of 1 cell thickness, and an outer spore sac which is 3 or 4 layer thickness. Between the inner and the outer walls is the cavity of spore sac. Now this spore sac, it contains numerous spores at maturity, but there are no elaters present. The third region of the capsule is the operculum or the lead. It consists of greatly thickened surface layer which is known as the epidermis. Now the inner wall is of 3 to 4 layers and it is small, thin-walled parenchymatous cells. Now this forms the major portion of the operculum. Underneath the operculum, there is a layer known as peristome. It is a long, conical teeth-like structure which consists of 16 teeth in each set. Now total being 32. The peristome teeth close the opening of the spore sac and thus it plays the important role in the dispersal of spores. So these are the references. Thank you.